So the next session is going to build on what we just heard about. This is going to be the complex molecular interaction session. We're going to have 4 talks. These are pre-recorded talks, so we'll be playing them out. And then at the end of each talk, we will have a chance for questions. So please feel free to start posting your questions during the talks so that we can go through the Q&A as fast as we can and do as many questions as possible. So I'll start by introducing our first talk. The talk is titled Mapping Cell Structure Across Scales by Fusing Protein Images and Interactions. And this will be presented by Yue Qin from UCSD.
Hi, everyone. I'm Yue Qin from UCSD Editor Lab, and today I'm going to talk about mapping cell structures across scales by fusing protein images and interactions. This work is also posted on bioRxiv, so check it out if you're interested. Biological systems are naturally organized into different scales. If you think about it, like you start from proteins at a few nanometers, and they bind with each other to form complexes that tens to hundreds of nanometers, and then form pathways, organelles, and all the way up to cells that are often at a few micrometers. And today I'm going to show you how I'm able to infer a multi-scale structure by fusing the data from largely different scales, and namely the affinity purification mass spectrometry data that captures protein-protein binding information at a few nanometers, and confocal images that capture the overall global protein localization that's often like at micrometer scales. To start with, so in this work, we restricted ourselves to use only data from the HEC293s in order to control for the variations caused by biological context. And this also ensures that we limit the noise to ensure that only valid and stringent conclusions are being drawn in this study. And more specifically, the data sources that we used for our image data were from Human Protein Atlas and the protein-protein interaction data from APMS experiments are all from the Bioplex database. And together, these 2 databases give us 661 proteins that have both image and protein-protein interaction data. In order to extract useful information about proteins from these 2 different types of data modalities, what I did is I used a deep neural network in order to extract relevant features out of these 2 types of data. For image data, I used a deep neural network to extract for each protein a 1024 dimension vector that captures the global protein localization information and also like the relative localization information of this protein regarding to the cellular markers such as nucleus and cytoskeleton. And for protein-protein interaction data from APMS, I use another deep neural network to extract another 1024 dimension vector that captures the binding partners and the interaction neighborhood of this protein inside the data. So just to clarify that each protein has 2 1024 dimension vectors, one capturing the information inside the image data and one capturing the information inside the APMS data. Before we go on and integrate them, it's also important to reemphasize and point out that these 2 data are capturing information that are of very different physical scales. So like if you directly compare them, what you will able to see is a lot of noises, and it's just very hard to separate signal from noise. So that is why before we integrate, we need to find a way to best coordinate these 2 kinds of data to extract the most useful overlapping information out of them. The way we do that is by mapping these 2 kinds of data first onto the same physical scale. And this is enabled in our study by the observation that you can actually use gene ontology annotation to approximate distances in nanometers. So what we found is that for cellular subcomponents such as nucleolus, lysosome, centriole that's labeled on the graph here, the actual physical size documented in the literature is positively correlated with the number of proteins this corresponding system has inside gene ontology. With this observation, we are able to derive mapping rule in our study that can translate the gene ontology of each, that can translate the annotation of each protein inside gene ontology to the pairwise distance between them. And using this curated distance, that is how we're able to calibrate the relative distance between each pair of proteins at the cell given the features that we previously extracted. And this is done by a random forest regression. And on the right, I show you the performance of our random forest model. So it clearly shows that our random forest model effectively learned the relative protein information from the features that we extracted compared to the random features. And next, after we have a relative pairwise distance between all pairs of protein, we're able to apply pan-resolution community detection to finally get a multi-scale subcellular hierarchy. This pan-resolution community detection was done by gradually thresholding on the relative distance. So what we did is we've first only look at the most close, the protein pairs that are most close to each other inside the cell and what of community that they form. And then we gradually relax this distance stringency to bring in more proteins that are not that closely associated with other proteins. Then this gives us larger and larger communities. And eventually this will include all the proteins, and that is the root node and also the cell. And this is how we're able to construct the multi-scale integrated cell, which we call MUSIC version 1.0 inside our paper. MUSIC resolved 69 subcellular compartments, and of which half are previously undocumented. And for those, we call them putative systems and are labeled in purple here. So we next ask is, what, like for each system, exactly what data is contributing to its formation. And the way we do this is we study, like for each system, when is it most robust, like to what data is it most robust. So what we found is that imaging data tends to robustly identify larger systems, while APMS data, on the other hand, robustly identifies smaller subcomponents that are within the image systems. And moreover, the highest robustness is observed when you integrate both image data and APMS data altogether. We next validated MUSIC by doing additional APMS experiments, and this is done by our collaborators from the BioPlex team, Ed, Steve, and Wade. And what we found is that MUSIC is significantly, a lot of systems in MUSIC is significantly enriched for the new protein-protein interactions that are identified in the additional APMS experiments. And more importantly, 64% of MUSIC systems can be validated this way. Here, I'm going to show you one biological story that can be inspired by MUSIC. Is that MUSIC identified a novel protein assembly of 7 MUSIC identified a novel protein assembly of 7 proteins. And here I'm showing you the images of these 7 proteins on the left. And by interrogating the function, the already known function of each member of this protein, we found that a lot of proteins are participating in pre-ribosomal RNA processing. And that's why we named this system pre-ribosomal RNA processing complex 1. More important, so originally in the input data from BioPlex, these 7 proteins have no known protein-protein interactions among them. However, with the new APMS experiments that we performed, we found that actually majority of the protein pairs within this system are physically interacting with each other. And more importantly, this interaction, it is enriched within this system because we observe a lot more fraction of protein pairs that are physically interacting within the system compared to the protein pairs that are between this system and its parent system, or between this system and all the other systems inside music. And for this, I want to thank all my collaborators, and this work is supervised by Dr. Trey Addiker, my direct supervisor, and Dr. Emma Lundberg from Human Protein Atlas Group. And also want to thank Jason, Fan, Jisoo, Adriana from Addiker Lab, Jianzhu Ma, who is now assistant professor at Purdue University, and Casper and Wei from Emma's lab. And I want to thank Wade, Steve, and Ed for the APMS data and support. And I want to thank my funding sources from NIH and my program, Bioinformatics and Systems Biology at UC San Diego. And thank you all for listening.
That was a great talk, Yue. So let's just wait for a few questions. All right, so we have one question from the audience. You mentioned that a challenge for doing this in many cell types is that data for not many proteins is available. Have you considered using transfer learning, like pre-training on HEC293 or multitasking to try to do this on more cell types?
So in this study, like the deep learning method that we use for the image embedding, it's actually applied, like it's actually applicable to many different cell lines. And the reason that we're currently doing this only in HEC293 is because we're trying to investigate the relationship between the imaging data and the APMS data. And so, in order to derive the convincing conclusions as kind of a preliminary study to like in the future, of course, a whole proteome cell map and in multiple cell lines, we need to make sure that our method is able to really capture what we're thinking is captured. And this way, so that's why we excluded, including other data sets and other cell lines. And so the deep learning method, as I mentioned, for the image embedding, it's already being trained in all the cell lines available in the Human Protein Atlas, and it's applicable to all of them. And also, like for the APMS data, we use the Notuvec, and it's also applicable to various different networks.
That's great. Just following up on that question, if you had data across multiple cell types, would you train separate models for each of them, or could you train something jointly across all of them?
So as for the random forest embedding, for the random forest regression, I do think you can train jointly, and because it is just learning this protein distance based on the features extracted from data. Yeah. However, for the final multi-scale cell mapping, we do think it's necessary to build a different cell map for different cell line to better observe like the differential protein communities that's formed within each cell line and to study cell type differences.
Great, awesome. One last question. So do you think, is there a specific reason you picked the random forest as your last sort of your integration model? Would it be possible, had you also tried using just a fully connected neural network or something of that sort?
The reason we use random forest is because it's kind of the first try for a non-linear way of integrating the data. And we haven't tried a fully connected neural network because we already observe a good performance in the random forest. So I think we don't need to go like to a more complicated form of training, but it's potentially something that we could try.
Great. So I don't see any more questions. So I think we'll move on to the next talk.